Okay, welcome everybody. I think we can start. So today's SPAAMtisch seminar will be given by Giulia Zampirolo, who is a postdoc at the University of Copenhagen, and she's working with ancient environmental DNA from marine cores. And she previously worked during her PhD on rock shelters deposit in Czech Republic, which is also the project she will present today. So Giulia, I give you the floor.
Yes, thank you. I will share my screen. Can you see it?
Yes.
Good. Just check if this works. Okay. Yeah, so hi everybody. I'm Giulia and thank you for inviting me to this workshop. Yeah, so I will present today one of the projects I've been working during my PhD, and it was published as a preprint, and we had good positive reviews back from Current Biology. So hopefully we can publish soon here. Its title is Early Pastoralism in Central European Forests, Insights from Ancient Environmental Genomics, and it represents a study, a collaboration with archaeologists and paleoecologists from the University of Calgary and the University of South Bohemia. So we are actually in Czech Republic in a sandstone landscape, as you can see from the picture here. And one of the key questions of this study is the process of neolithization in Central Europe. That's still an open question. So in the Bohemian forest, we have hunter-gatherers during the Mesolithic, late Mesolithic, managing the forest in the area. And we have early farmers arriving in the area 7,500 years before present that were occupying the lowlands. And we have some rock shelters in the forest. And these rock shelters were accessible both from to hunter-gatherers and to the early farmers. So this environment represents like a peripheral environment to both ecotones. So we actually are in the Český rainforest in northern Bohemia. As you can see, the green triangle in the rock shelter is called Velký Mamuták, and is at 5 kilometers from the closest Neolithic site in the area, and it has 10-meter overhang. And this overhang is formed by Cretaceous sandstone. And there has been an open trench in 2018, as you can see here from the figure. And there was a previous study based on paleoecological remains, where actually is the Pakova et al. Study in 2021, where they built some age depth model. And you can see that the occupation of the area started in the Mesolithic, and they have some remains up to the Iron Age. So it has a long occupational period. There were some analysis on pollen data, but there was kind of a scarcity of remains for the Neolithic period and the early Bronze Age period. But there was some fragmented bones that were found through whole the period, and some coprolites that was suggested to be connected to some goat or sheep. And the most abundant layers are the one from the Bronze Age and Iron Age. So it was quite a big question what was happening before that period when the farmers arrived to the area. So what we wanted to do is to investigate this deposit through metagenomics. So my supervisor, Mika, took some sediment samples in 2018 on the profiles of the archaeological trench. And we want to investigate what was the herding practices and if we can say something about the forest management during the Mesolithic and Neolithic period. So what we applied the shotgun sequences of the entire metagenomes to try to have a taxonomic classification of our genetic reads and maybe do some phylogenetic and population genomic studies on this. And this method, of course, has many challenges because it's highly dependent on the quality and on the availability of good reference databases. So we actually took sediment samples of around 0.2 grams and that were manually processed in the labs at the Geogenetics Ancient Lab in Copenhagen. We extracted DNA and we built double-stranded libraries. And then we sequenced with shotgun sequencing. So all small fragments of DNA were just amplified with the Illumina platforms. And we applied some bioinformatics tools to clean and analyze our data sets. So we used the Holy pipeline that's my supervisor developed and is in the one I've been working on during my PhD. So we remove adapters, we remove duplicates, and we map to the existing databases. We do some taxonomic classifications and authentication of post-mortem DNA damage using the tool that's called MetaDamage, and that's still under development at the University of Copenhagen. And I will introduce you to this tool because I think it's quite a key method that is used in this study. So MetaDamage is actually a tool that is based on the assumption that there is chemical damage that accumulates over time. And this damage can be quantified and qualified and based on the C2T and G2A transitions happening at the on forward and reverse strands of the DNA. What this tool can also do has been implemented with the NGS LCA algorithm, so to assign different taxonomical ranks to the reads. And so the damage can be qualified and verified for different ranks. So genus, family, yeah. And one of the main output from the tool is actually a damage profile, where you can see on the y-axis we have the percentage of damage, and on the x-axis the positions through the strand, and you can see how the level of damage is how it's supposed to be for ancient reads. It increases close to the ends of the strand, and this model that's where this tool is based is a beta binomial model where actually also the certainty of damage is calculated. So how much we can be sure that this read contains that level of damage. And you can see from these plots, it's from the Mikkelsen et al. Paper, and that will still a preprint in 2022. So we have on the y-axis, the level of damage, and on the x-axis, the significance of that damage to be true. And you can see on a simulated data set how ancient DNA actually appears to have higher significance and higher level of damage compared to non-ancient DNA. So applying this method and with the implemented with some good knowledge of the geological evaluation of the deposits and the presence of radiocarbon dates. So to build an age depth model, we can actually try to track the level of the damage on the full data set, on a metagenomic data set through time or through depth. And this is one of the figures I had in my the supplementary of the preprint. what we can see in the first plot is just the different defeat. So in the x-axis, you can see the degree of damage that's reaches 20%. And then we have the depth on the y-axis. We can see how we have higher level of damage, so low to high level of damage. And then we applied some filtering, some thresholds. And this is still a process that's under development and that needs more standardization because at the moment, the thresholds we apply are data-driven thresholds. And as you can see, we also lose some data applying these thresholds. So we have kind of balance, what we're looking for, what is ancient, what we can include or not. And that's one of the biggest challenges actually of using this tool. And another tool I applied is Pathfinder, and is actually this tool allows the placement of ancient DNA reads from a metagenomic data set to 3 branches, analyzing the single nucleotide polymorphism, or what you call the polymorphic sites in the genomes. So giving a likelihood based on the counting of these polymorphic sites, it provides a number of uniquely supporting SNPs or uniquely conflicting SNPs, and place your reads on a tree, on a phylogenetic tree. This is one of the tools I was using to verify the haplogroups of some of my reads that we'll see later. So going directly to the results. So here I present the animal DNA abundance. We have on the y-axis the timeline from Mesolithic to Iron Age, and then we have on the x-axis the percentage of reads, proportion of reads that were actually mapped to the different genuses. In this case, for animals, so metazoans. So we can see that, for example, we have goat, only goat actually from the Neolithic layer, 7,000 years before present. And unfortunately, we could not say if this was a domestic or wild goat. We have the full package of domesticates, starting from the late Neolithic, where we have cattle and sheep. And actually, sheep is one of the most abundant taxon in our data set for the late Neolithic period. If we look at the Bronze Age, instead, we have a really high abundance of pig or boar. And this is a period when actually matches with archaeological remains, where there was also a high abundance of domesticated pigs in the area. Yeah. And we also find some human DNA for the Bronze Age period. So what I did actually, I tried to extracted the reads that were part of the genus of the different domesticates and tried to map them to modern reference genomes to verify if it was possible to define their haplogroup of origin. And we could do it for the sheep, for the late Neolithic period. That appears to be an ancestor of the haplogroup B or European Mufflon. And we could do this also for the Bronze Age period with the reads mapping to the cattle. So I just, and these haplogroups are connected to the haplogroups that actually were brought to Europe from the Near East by the early farmers. So actually they match also the archaeology and the genetic history in Central Europe. So what we did, this is just an example, show how we did this type of analysis. We ran a BEAST-3 with the modern reference genomes, in this case of sheep. And we extracted our reads and mapped to those different haplogroups. And then we ran the phylogenetic analysis with BEAST, and we can see how the ancient reads are sister clade, a sister branch of the Ovis aries haplogroup B and the Musimon. So the 2 haplogroups I was mentioning before. And then we run Pathfinder to check how much. So we can count these polymorphisms and can replace and see how many supporting polymorphisms we have in the mitochondrial genome. Then we also wanted to try to track what was the sources of our microbial DNA using Source Tracker 2. And we had help from Antonio Fernandez Garra, who came recently to University of Copenhagen. And we can see that the ancient communities of bacteria kind of match with the animal DNA abundance. So we have a higher proportion of gut and fecal microbiomes of domesticates coming from the late Neolithic period, with high abundance of sheep and fecal and bovine, rumen and fecal microbiomes. And we have a higher proportion of pig microbiomes, and we have some human also fecal microbiome from the Bronze Age period. We also analyze the plant DNA abundance, and we can see how there are some key taxa that are connected to, that are high nutritional for bovines and are maple, ash, and elm. And we can find them for Neolithic and Bronze Age period mainly. And this can be connected also with some studies in the area where actually woody plants were used as winter supplement for livestock for the nearby sites in the lowlands. So it's kind of consistent with this remains. So to sum up, we have animal DNA to elucidate herding practices. The goat in the Neolithic could be interesting to have further analysis on this, because it could be super interesting to know if was really managed by, was just wild goat. But we have domesticates from the late Neolithic, and we have what it looks like a transition of herding practices during the Bronze Age. How much this is due to DNA preservation? Is DNA preservation a key issue? We had a key issue in the Mesolithic because unfortunately we could not retrieve enough DNA. Is that connected to DNA preservation, or they just, humans were not there, were not keeping livestock there? We have our analysis on the bicromial communities. And is this tracking the origin of DNA? Can we say that at least partially DNA comes from the coprolites that were also found in the dataset? And if this is true, the plant DNA we found can be highly connected with the feeding the livestock. So of course I have to thank all the people who were working with me, the archaeologists and my supervisor. And I've just put some pictures of a trip we did in 2022, where actually we were looking around rock shelters in northern Bohemia, and we were just finding new possible sites to analyze. And thank you very much for your attention. Grazie mille. If you have questions, I'm happy to answer.
Sorry, I didn't see who rose the hand before, between Bianca and Nikolai.
No, Nikolai can go first.
Ladies first.
Okay. So I have some questions, especially about the plant part. So I was curious to know about the database. If you have, if you were using the whole database, or if you built some kind of custom database for plants with the specific like, let's say, added some specific taxa that you thought they could be in that, let's say, region? And also, at which level did you do the assignation? And another one, then I finish with the plant part. So, and how did you calculate the abundance of plants that you showed?
Mhm.
And for the microbial part, I don't know. Of course, like you said that Antonio helped with it, so I don't know if it's a question that either makes sense or that you can answer. But so I was curious about how to decide the sources for the microbial part, because it's something that that we are dealing with now, and so like we tried, and so I see that some of the ones that you chose that have been chosen were the same, but I'm curious, like, if it's based on that environment or it's a more general rules on, you know, soil sources.
Okay. So, wait, I will go back so maybe it's
Yeah,
easier.
sorry.
So for the plants, we didn't use custom databases. We used the Chloroplast database. I believe was also Philo Norway database in it. We used all the database available. We didn't do any custom-based database. The level is the genus level in this case for all the different, so also for animals. Then the abundance is just, I just show the proportion of reads.
Okay.
So there is no calculation there is no or rarefaction or any type of.
Okay.
It's just the proportion of reads that we get from the layer.
Okay.
And you can see like these small numbers are actually the number of reads.
Oh, breeds. Okay. So, and do you have a
Probably here they are
threshold?
not. Yeah, you're right. Here there is no number of reads, but it's exactly the same. So just number of reads.
Okay. And do you have a threshold that you decide that, you know, you consider, okay, it is from this taxa, or no?
What do you mean?
Like, I don't know. Well, in your case, it's alignment based, right? So,
Yeah.
okay. Yes.
Yeah.
No worries. I answered myself.
For the microbial, we selected the sources based on what was our question. So we knew that we had like animal DNA. So we selected like, okay, let's do bovine, let's do sheep, pig, human. So the main gut, fecal. We also ran some tests with urine and other types of environments. And then for the the different environment, you see like temperate forest. So we wanted to see, okay, can we track temperate forest? Can we track wetland, grassland? Yeah. So we just try to guess based on the environment we are analyzing.
Okay.
Yeah, but then we had some like kind of negative control. So using like other human stuff and using like how is called agricultural soils.
Okay.
Yeah. But it's mainly like within other soil, like you see the gray, like other soil is like other environments that were not part of this area.
Okay.
Came out like other soil. And then we had a lot of unknown because probably there is no data for that, or I don't know. So but it's quite complex. Yeah, I believe it's one of the main key points, like how to do this type of analysis. Yeah.
Yes, I agree, because especially with this, you know, with soil and with microbes in the soil, a lot of them also they're not yet known. So that's pretty tough, but it's super interesting to see that someone is, you know, doing it and trying to do it. So thank you so much. Really great. Nikolai, you can go.
Thanks. Yeah, hi, Giulia. Thank you very much. It was a nice presentation. I was about to lower my hand, actually, because I just realized, looking at this slide, that please, yeah, I just wanted to double check with you. So you're saying you found cattle or goat based on the fecal samples, right? Not from the soil. So I maybe misunderstood. So did you catch this animal DNA from the soil or from the fecal remains?
From the soil. So we just got soil samples. So
okay, then my question is still valid. So basically, yeah, I myself found sort of cattle and a goat in many metagenomic samples where they were supposed to be present and not supposed to be present. So basically, what I learned is sort of it's kind of you sometimes see some traces of animals in certain samples, but then the challenge is actually to prove that the DNA you think belongs to cattle is actually a cattle DNA, or sheep belongs to sheep. So basically, I'm curious, sort of how you prove that sort of it's not a reindeer, it's not a mouse. You know, maybe you can show sort of numbers of reads, how they're spread, how many mismatches they have. Maybe you had negative control samples. So basically, the challenge is actually not to catch an animal DNA, but to prove that this DNA belongs to the animal you think about. So that's kind of my sort of obstacle that I'm struggling all the time. So I wonder how you prove that this DNA comes from goat and not from a reindeer.
Yeah. So one of the things that we did is like remapping these reads to the mitochondrial genomes. So in this case, you can kind of investigate if you have mitochondrial genome there, and that can be a second step where you say, okay, we do have that too, and it aligns to, you know, some species or some general haplogroups, as I said. So in that case, you can try to authenticate it in that way. But of course, you need to have enough reads also to do that. So some deep sequencing or targeting also. So I think in this case, they are authentic, especially the ones that we could double check with the mitochondrial phylogenetics tree that we could build. Of course, is difficult, you know, I think it's one of the main challenges of metagenomics, for sure. So that's my idea, at least.
But if you go to mitochondrial reads, are they kind of more specific? Or like
yes, because in this case, actually, with the algorithm we use for taxonomic classification, we stop at genus level. And we think that the genus level is reliable enough, not at species level though. So to go at species level, we have to do further analysis. And in this case, it's the mapping to the mitochondrial genomes. Yeah, but I think one of the problems maybe is with the rarest taxa, right? The ones that have lower abundance, because in that case, how can you authenticate those ones? So maybe those are the main issue in this case. And also, as far as I understood, when I don't know which tools you're using, I'm not sure. I'm also not a bioinformatician. Actually, I come from archaeology, so I learned a lot during my PhD, but the NGS algorithm we are using is kind of detecting which are the unique sites full genome. So in some case, when you reach family or genus level, you can kind of be sure that's unique of that genus or that family. Then from those reads, if you need to go farther down to the species, we extract those reads and we try to map to the mitogenome. That's what we try to do.
Yeah, just last question. So when you say you found a goat, so is it based on 5 reads or 500 reads
In
or
this
5
case,
million reads?
In this case, it's not much actually, it's 500. You see down here, like small number, 568 reads. We try to map to the mitogenome, and we are very low mitogenome, so we cannot say the species, we cannot say, but we believe it's true, you know? We believe it's true because we know that the archaeological also data set says that. And
Yeah, I'm just
Martin.
basically thinking about that this, yeah, if you take a reindeer genome, there will be a few hundreds of reads mapping to reindeer as well, probably. That's
Yeah, but it's yeah, I understand that. But when we do the mapping, like we use all the different data sets, and it's based on the alignment and on mismatches. So the ones that have they are more similar, the reads that are more similar to the genome, they are kept. The ones that are not, they're not. And then during this algorithm, they there is identification of the different assignments, so family, genus level, order, blah, based on these transitions and transversions in the genome. So they kind of check the unique transition.
I'm sure they're ancient. I'm sure they're ancient. I'm just questioning, or sort of that's at least my challenge, is to actually prove that these reads, the ancient reads, they can be ancient. They just come from a mouse, you know. That's my sort of a challenge to kind of really
I don't know if I
demonstrate these are the goat reads.
Can I help you more? I don't know. Are you, if you want me contacts, I don't know. We can have a look at your data and we can help you. I don't know, in some way, maybe. If you want me to have a look at your data, just.
Yeah, I think I got some
reach out.
idea how you're doing this. Yeah, thanks.
Freya.
Thank you. And thanks for the great talk. My question also aims in pretty much that direction, or at least one of my questions. And that is, if you are able to identify cattle like this, if you would have wild species also roaming in your areas, would you, with your method, be able to distinguish wild cattle from domesticated?
Yes.
Yeah.
If you have enough reads, yes.
All right.
So yes. So
if you have enough coverage of the
exactly.
Okay.
In this case of the mitogenome. So what I suggest to you is if you want to have a study on I don't know, from the sediments, for example, coprolites, and your question is specific to cattle. In this case, maybe metagenomic is not the best way to do it. Maybe you should do some targeting. So you can target specific part of the mitogenome that you want really look for. You know, in that case, you can then see what do you get if you get domestic or wild. So then you can try to build a phylogenetic tree, place those reads, and see where it goes. Is it more close related to the domestic or to the wild?
Yeah, and you even found the haplogroups to.
Yes, we try to build based on haplogroups. Yeah.
All right, and my second question would be you had a slide with the depth of the findings and the damage quantification, and there I would be very interested in. I'm not sure if I had a look close enough, but is what I'm seeing also a little bit of an effect that in in very young, of course, in very young layers, the damage is not as progressed. And then there is one area where it's highest, and then more layers on top maybe also has an effect of preserving, like and at one point of depth, there is a preserving factor coming into place.
Yeah.
And is that here around these 1 meter 30? And is that something that is unique, you think, to this site? Or can this be generalized on cave sites with similar?
You mean the range of depth, or
What I mean is when the damage of the DNA is progressing due to time.
Yeah.
I mean, you have here, of course, the factors time and depth. And if there's, or what is the point of optimum preserving? Is it, or no, I mean, the other way around, maybe. What is the worst case for DNA? Is it to be, yeah.
So there are many factors actually that are connected to DNA preservation. So one of these is temperature. So for example, here you are in temperate environment, and it's not like the cold Greenland, you know. So there you can have way better preservation of DNA compared to this site. But in this site, we can have better preservation compared to tropics. So temperature is a key factor. Also the soil. So the texture of the soil. Clay looks like to be the best. But still, it's something that people are still studying on it. And it's not something that, okay, it's like that, you know. So I've applied this tool to other different deposits, and you can see how it can really change the range of damage based on the type of deposit you have. So for example, the 2 million year old paper, if you had a look at that, where they took sediment from Greenland, like 2 million years old.
Yeah, I know that one, yeah.
Yeah, so they have around like 40% of damage, 50% of damage in layers that are 2 million years old. So it's like, okay, huge, like a lot of preservation, good preservation. But here we are in temperate environment and we have maximum 20%. So it looks like it's way more recent, you know. So we expect to have less damage, but still, it's still a lot, you know. So it's something that we cannot say. It's something you can just you just need to verify with your deposit how it is.
Okay,
But,
yes.
sorry. Yeah, but the general trend is that like you have an increase of the level of damage through time. So that's something we see everywhere.
Yeah,
How
exactly.
much?
That is something we see everywhere, but I would be interested in the point where the depth turns over as a positive impact on the damage.
So in this case, you mean that, for example, the bottommost layers have lower level of damage. So in that case, what I think it's connected, but I don't have the, you know, golden answer. But I think it's connected to the fact that we cannot retrieve. So it's the sample maybe that was not good enough. We cannot retrieve enough reads. And so in that case, we only retrieve reads that have lower damage or that are more modern, you know. So it's like we don't have it, you know, we couldn't get it. Maybe these layers needs targeting or needs some other methods to get DNA out of it. So it's challenging because we expect to have higher level of damage there, but we don't find much reads there. So maybe it's because we don't have reads there, we don't have enough DNA preserved. I don't know why. I cannot say why.
All right, thank you. Yeah, I would have thought that maybe like the greater the depth because of more sediments growing on top of it, then more stable temperature, less impact of humidity, and general better preservation. But if you say that there are also not a lot of reeds from those layers, that of course explains it very differently, right?
Yeah.
Thank you for your explanation.
Yeah. Welcome.
Maria.
Thanks for, can you hear me?
Yes, yes.
Okay, amazing. Sorry, I haven't used these earphones before on a talk. Thank you, Giulia, for a great talk and a really exciting project. My question was about the same graph. Do you have radiocarbon dates for your different layers in the?
Yeah, we do.
That's really cool. It would be nice to correlate your damage patterns through time also with the dates and the paleo-environmental conditions. I've been wondering actually about DNA preservation through time, if the conditions at time of deposition itself is more key than, well, or has an effect that is non-negligible over like the age sometimes. If, for example, it was deposited during a colder, drier time period, that sort of damage degradation, which happens mostly in the very recent time post-deposition and then slows down, is kind of like has less, is smaller, and then it's deposited and it's more stable than some things that might be deposited in more shallower layers where it was more temperate. And so I was very curious looking at your graph on the left, but if you're telling me now that that graph also doesn't have as many reads, maybe this isn't the specific answer, but it would be really interesting. I don't know if you've already done that in your paper, just looking at
we didn't
temperature and
no, we didn't do that. I mean, I can show you the study they did before. Sorry, to go back, which was the time range that actually was. So yeah, so these are the radiocarbon dates connected to the different periods. Yeah, and yeah. I didn't look into the different environmental conditions through whole the Holocene. So our key points where we didn't find DNA was these ones actually. So the Mesolithic. So during this time, 10,000, 9,000 years before present. Yeah, maybe it could be. It's a good hint. Can be an explanation.
Yeah, I think I'd really like to look into that in my own future work as well.
Yeah.
It would be nice to know exactly what affects things in preservation.
Yeah, that's good. Good key point. Yeah.
Thank you very much.
You.
And again, well done with all of this work.
Thank you.
Yeah, I have a last question, Giulia. Actually, two maybe. So one's always related to the damage. Like, since you are looking at so many species, right? No?
Yeah.
You like non-human, human, and plants, and like, do you see? Do you think that the damage patterns is always how do you take as a reference always the human damage pattern to like assess whether a reed is damaged or not? Because I mean, I observed that some microbial species, they may not show the damage, so we are sometimes maybe wrongly discarding them, even if they are authentic. So like, have you observed, say you had an ideal condition, like normalized for, I don't know, the depth and the age, and did you observe like this kind of fluctuations among species, or?
So, yeah, so it's not something that's part of this study, but since we are developing some models to analyze the meta-damage output with all different things we have, right? So microbes, plants, animals. We do see a pattern in which there are some species-specific things going on. So looks like plants are have a different rate of degradation. And maybe it's connected to their different biological conditions, like how the DNA is, how the cell is. So probably biological feature of that. For the other things, I think we didn't look into, especially into microbes.
Yeah.
I guess would be a lot of headaches with that, actually.
Mhm.
But what we're trying to build is some standardization, so some models based on the plant data.
And did you discard the microbes that you thought they may be authentic but without damage, for example?
In this case, yes, we
Yeah.
had to be conservative. So the ones
Even if they were like in very deep layers, for example, if you have many checkbox that you are quite confident that these are like water.
if they looked like ancient, we kept. If they didn't, we removed.
Right.
Or
Okay.
if we were unsure, we removed because
Okay. Mhm.
yeah. So we are super conservative, you know.
Okay. And then my second question is more about because you mentioned a molecule. You mentioned beast, so I'm not sure if you did molecular clock, like
We didn't do
estimate.
molecular clock.
Okay.
No.
So you used it just to estimate the phylogeny.
Yes,
Okay.
exactly.
Okay, I
Yes.
see. Okay, so no question. Okay, thank you.
Yeah.
Any more questions? Okay, so I guess if that is all, thank you, Giulia, for participating. Thank you, everyone, as always, for participating. And we'll see, I mean, for the next month, people please volunteer.
Yes, and I remind that the recording is gonna be uploaded in the
Yes,
YouTube
yeah,
channel, right?
if you ask permission, then we can upload this recording to the YouTube channel.
Okay.
Okay, so